लाईव्ह स्टार्ट केलंय.
Hello. Good morning, one and all. On the behalf of Rasashastra and Vaishajya Kalpana Department of Dr. G.D. Paul Foundation's YMT Ayurvedic Medical College and Hospital, I, Dr. Ashwini Deshmukh, Associate Professor in the RSBK Department and also coordinator of today's webinar, warm welcome you all. Before proceeding further, let's pray to Lord Dhanvantari on this occasion. I request Dr. Padmaja to chant Dhanvantari Stavan.
Namaskar. Namami Dhanvantarimadidevam Surasurairvanditapadpadmam Lokai Jararu Bhayamrutyunasham Dhataramisham Vividhaushadhinam Shankham Chakram Jalokadadamrutaghatam Charudodvishchaturbhi Sukshma Swachhatirudyam Shukaparimilasal Maulimam Bhojanetram Tarambodojvalangam Katitati Vilasan Charupitambaradhyam Vande Dhanvantaritam Nikhilakadavanam Praudhadavagni Neelam.
Thank you, Dr. Padmaja. Now, I would like to call upon Dr. Sheila Pargunde Madam, Professor and HOD of RSBK Department, for delivering welcome note. Over to you, ma'am.
Yes. Good morning, one and all. Namaste. I, Dr. Sheila Pargunde, organizer of this webinar and also Professor and HOD of Department of Rasashastra and Vaishajya Kalpana, Dr. G.D. Foundation's YMT Ayurvedic Medical College, Hospital and Research Institute, Kharghar, Navi Mumbai, warmly welcome you all in this webinar 2022 organized by Department of Rasashastra and Vaishajya Kalpana. It's my privilege to welcome our guest speakers, Dr. Sadhana Sathe Madam and Mr. Rajendra Tugnar. I cordially welcome Honorable Chairman Dr. G.D. Paul sir. He is a source of our inspiration and motivation. I welcome respected Dr. Sanjeev Yadav sir, Dean of our college, and who also support and guide us in such academic activities. I heartily welcome respected Dr. Ninath Sathe sir, Vice Principal and also Professor in our department, for his kind support and guidance for this webinar. I also happily welcome Dr. Zaikini Madam, Research and PG Coordinator in our college. I wholeheartedly welcome our delegates. I heartily welcome our sponsor, Pillcraft Advanced Remedies. I warmly welcome our faculty, Dr. Ninath Sathe sir, Dr. Meenakshi, Dr. Ashwini, Dr. Vaishali, and Dr. Ashish. I also welcome our UG and PG students. And last and not the least, I also welcome our non-teaching staff. Okay, thank you.
Thank you, ma'am. Now we will proceed to first session of today's webinar. Again, I request to Dr. Sheila Pargunde Madam to give brief introduction of our first eminent speaker, Dr. Sadhana Sathe ma'am.
Yes. Hello. I would like to introduce our eminent speaker, Professor Sadhana Sathe Madam. Dr. Sadhana Sathe Madam is an eminent research scientist working in the field of pharmacology and toxicology with an academic experience of 31 years. The overreaching field of her research involves various neurological disorders like epilepsy, Parkinson's, and Alzheimer's disease, as well as metabolic disorders like diabetes mellitus and related complications. The research focuses on establishing novel therapeutic interventions by exploring herb and modern medicinal drugs. She has mentored several graduates, postgraduate, and PhD students to pursue a successful career in pharmaceutical space. She has over 116 national and international publications to her credentials and has been a renowned speaker in several esteemed national and international conferences. Her horizon is not limited to academia, but also as a consultant to the global pharmaceutical industry, where she extends her services in regulatory toxicity studies and pharmacodynamic evaluation. In addition to her scientific pursuits, Professor Sadhana is a fitness enthusiast, accomplished yoga trainer, and enjoys outdoor adventures. And she is professor in the Institute of Chemical Technology, Mumbai. Thank you.
Thank you, ma'am.
Yeah.
I request Dr. Sadhana Sathe, ma'am, to start the lecture.
Okay. Good morning, everyone. And I thank the organizers. Can you hear me?
Yes, ma'am. Yes,
Okay. Okay. So, good morning, everyone, and thank you so much for inviting me, first of all, and introducing me in such an extensive way. It's always been a pleasure to connect with YMT College's Pharmacology department with Dr. Pargunde, Dr. Ashwini. And it's my pleasure to present my talk today. And that's about 1 minute. I'll just share my screen. Okay. Can somebody help me? Because they say that host disabled participant screen sharing. So can you just...
Just a minute, ma'am.
Yes, yes.
Okay, now check it, ma'am.
Yeah, sure. It's still disabled. Not possible. Should I check it now?
Yeah, ma'am.
Okay, there. Okay. Who can share? Host only, all participants. So host only is still okay. I only did it. I'll try right away. Sorry. Yes. Can you see the screen now?
Yes, ma'am.
Okay. So my topic for today is preclinical evaluation, particularly pertaining to the experimental pharmacology. So even if I'm a pharmacist, I'm working in modern medicinal drugs, but it's always been my interest to explore the Ayurvedic drugs, herbal phytoconstituents, etc. So let me start my presentation. One second, I'm just having some issues. Yeah. So basically, to do the pharmacological experiments, you should have a basic research methodology. And that's what you all have been working towards it, where you study systematically activities of certain materials. In your case, it could be Ayurvedic drugs. So all that you need to do a study or to initiate a study is basically define a problem do literature survey, enough literature survey for whatever problem that you wish to explore. Then you design the study. After designing the study, the execution. So, most of the time goes in first three aspects of it: the literature, the defining of the problem, the literature, and the study design. And the rest too, meaning execution, takes about not more than 3 to 4 months, even if it is a extensive, difficult study. Statistical analysis will take very less time because of the available statistical softwares. And always remember that you have to even publish or patent whatever that you explore. If not patent, because for Ayurvedic drugs, it's a traditional knowledge and it may not be possible for you to patent a particular drug, but then you can patent a technology. If you make some formulations like eye drops or a vati or whatever is your formulations, if you could do that, you could patent those formulations. At least publish the data so that people know that your at least your work goes to the society for that matter. Now, considering pharmacology, there are various aspects of pharmacology, and currently molecular pharmacology is being well explored these days because of cellular and molecular biology. They have come, like they have been explored very well since 1980. Most of the drugs also are biotechnology drugs, and that's the reason molecular pharmacology is a very important aspect of pharmacology, molecular and biochemical. And if you see abroad, most of the experimental pharmacology is only about biochemistry and molecular pharmacology. Then the conventional pharmacokinetics, drug metabolism, toxicology, chemotherapeutics, that is drugs which are used in the treatment of infections, and various system pharmacologies that we know, like cardiovascular, etc. So main importance nowadays is given to molecular and biochemical pharmacology. So regarding the systems pharmacology, I'm just going through fast here because I want to discuss with you the case studies wherein you will understand better. So these are the different system pharmacologies that one can explore, whether it's to the brain or heart or gastrointestinal tract or respiratory system. And immunopharmacology has also been very important these days. You know about the COVID the epidemic that we suffered very recently. And also many of the cancer treatments, they evade the immune system, and that's why cancer drugs become resistant. So this is immunopharmacology is also becoming very, very what you say, popular. You can't say popular, but people are trying to explore that aspect of systems pharmacology as well. Now, regarding the methodology, it's three aspects: in silico, in vitro, and in vivo. Moreover, these days there's something more which is called as network pharmacology. Network pharmacology meaning whatever phytoconstituents or drugs that you want to explore, you just identify your target in the body, and you dock this target onto you dock many of your phytoconstituents onto this target, and you'll come to know whether your drugs are really active there or not. So you can screen many drugs using this network pharmacology. And this particular course, training course, is available at Dr. Bhushan Patwardhan's laboratory in Pune. So those interested students also think of this. This is really interesting. In a very short span of time, you can screen many of your drugs onto various targets for that matter. In silico is again completely software based in the computer, but a very valid and interesting way of screening drugs in a very short span of time at very least expensive for that matter. And then comes the in vitro, like you use animal organs or enzymes for screening your drugs, for evaluating your drugs, and then finally animals. So I'll be taking you through mostly the in vitro and in vivo assays, but I'll also explain you a little about in silico assays as to how we went about it. So these are simple questions. If you want, I can share this presentation with Madam Sheila, and she can give it to you so that you can really read it later. So these are simple questions that you need to ask yourself before designing any particular study. Now, this is a general information, like what are the biochemical classes or drug targets which are uh, uh, which are there currently today. So you can see that enzymes are the receptors are the major targets, 45%, enzymes are 28%, and then the rest. So basically, it is enzymes and receptors on which we could work on as our targets if we have to do in silico or even in vitro studies for that matter. Now, in an assay development, you read about it, you must remember that the assay has to be reproducible and it has to be cost effective. As much as automated, less personal variation and better results you can get. Now, biochemical assays can be done with enzyme receptors and hormones. We just saw that these are the three major targets. And cell-based assays, you can actually grow cells and you can check the cellular viability in case of anti-cancer drugs or cellular proliferations if you're using any hormones or tonics, or certain protein expressions or protein interactions or actual drug activities for that matter. So there are different in vitro or ex vivo assays. Ex vivo meaning you treat the animals and take the organs and then work on them. In vitro meaning you take the animal organs and work on them. So they are two different things. So in vitro assays, you have enzymatic, drug permeability, drug transporters, cell organ based assays, cytotoxicities, cellular and molecular biomarkers, bioassays, and antioxidant assays. So trust me, even these assays could give you a good indication of a antioxidant assays could give you a good indication of a drug activity. And other assays I'll talk when when I give you the case study exams. Now, one important aspect of pharmacology is pharmacokinetics. See, whatever effect that you are getting, whether you are actually seeing the levels of the drug in the body. Now, for your Ayurvedic drugs, it may be difficult because many times Ayurvedic drug is processed using various processes, or it is a combination of various herbs. So what you could do is you could just determine a particular biomarker meaning, supposing if you're using which drug I was ashwagandha is Videnia somnifera. So if you are using ashwagandha and if you want to see pharmacokinetics of that, you could think of vidinaloids, which you can see in the animal's blood for that matter. So you can design pharmacokinetic studies even for Ayurvedic preparations for that matter. And exactly like how modern medicinal drugs we do PK, you can do PK. Now, why should you include PK in your study design? This is just to show that whatever effect that you are seeing in an ex vivo or an in vitro model is not really, it's not a bias of yours, because the drug is achieving that concentration in the blood. That's why you're seeing the effect of the drug. So it's always better to see the pharmacokinetics as well as pharmacodynamics. And there are two aspects of pharmacodynamics. One is quantal. Quantal meaning the drug may work or may not work. Like if you're studying epilepsy, if you're studying epilepsy or fits for that matter, if you give a drug, whether it stops the fits or it doesn't stop the fit, that is one aspect of it. Or if you're studying blood glucose lowering, so is there any graded response? If you increase the dose of the drug, does the glucose level lower down in a better manner? So there are two kinds of responses, quantile and graded. I'll try to explain you as much through my case studies. So we have developed various in vivo models, meaning animal models. And I just love, because if you know the pathophysiology, and if you know a particular normal physiology versus disease pathophysiology, you can develop any model for that matter. So these are the different models that we have really developed in ICT. And I am okay. You can give me any problem, and I would love to help you in designing that model for that matter. It's very easy. You should just know the pathophysiology, normal physiology and pathophysiology, and what biomarkers could you take. That's it. That's what you need. Now, in a pharmacodynamic study design, when you want to see the effect of the drugs, you have to ensure that whatever animals that you get, they should be of the same strain, same age, same weight also, and from a supplier who is, whose colonies are good. Otherwise, recently I got one colony. I'm not going to name the person from whom I got it, but they all they all died, meaning they all suffered from some infection and died. Of course, the supplier gave me, replace the animals, but then your time is wasted, right? So, but ensure that the animals are good, healthy condition, and just call a veterinarian to see whether they are all good and in healthy condition, whether they intake good amount of food, water. So all these things are also extremely important while doing an animal experiment for that matter. And then materials, formulation, what is your formulation, whether it is a powder or a tablet or pill or a syrup or whatever. So when you give it to the animals, you have to ensure that you don't exceed the dose. Meaning, if the rat is only of 200 grams, you can't give more than 2 ml of a fluid into the rat. So that's very important. You should remember that before designing any study. And remember that if today I'm doing the experiment and my other student is doing the experiment, there will be personal variability. So as far as possible, you do your studies, you don't give it to anybody else, just to ensure that there's nothing wrong and you don't get any variability. See, what I'm trying to say is, if you keep all other things constant, then whatever changes is the drug administration, and then your results will be good. Now, I'm not going in the details of it. I will give you the presentation, but this slide means is that the blood levels and the pharmacological effect, they should match together. So this is called as modeling of PK and PD. And then this is the same thing which I have explained you as an animation, that the drug concentration, as in PK, should match with the clinical response. Drug concentration should match with the clinical response. Apart from that, many of times it is important to do toxicological studies also. So I had done one drugs toxicity studies, which was to be given over a long period of time. It was a rejuvenator, but it was an Ayurvedic drug, but they wanted to see its toxicity. See, many of the times the Ayurvedic preparations, they have heavy metals in that. Actually, that's not a problem if the Ayurvedic preparations are made in a particular way that the original text prescribed. But then the company thought that they need to do it, and we did the toxicity study for total 6 months for them. Now, when you do such toxicity studies as per the regulatory guidelines, then there is no holidays, no Saturdays, no Sundays, no festivals. If you start the study, you should complete the study as per the regulatory guidelines. So I'm just going over all these slides because I'm going to pass on the slides to madam, and you could read about these things. Now I'm going to go ahead with a case study which we did, and we did diabetic study, because diabetes is the most prevalent lifestyle disorder currently, And so what did I do? I did literature survey. I just found out statistics, what is the prevalence worldwide, what exactly is the pathophysiology, and what are the associated complications. So nephropathy, neuropathy, retinopathy, cataractogenesis, peripheral vascular disorders. And I have worked almost with everything except for peripheral vascular disorders. I've not worked with that. Uh, so this is about diabetes. I'm not really going into it because this is basic information which is available at any Google website. I did it from WHO. Now, what I want to tell you is regarding the pathophysiology of diabetes. See, normally what happens, you have blood sugar level between 70 to 100 when you're fasting, and when you eat complete meal, the sugar level should not exceed 150 milligrams per dl. That is for sure, that's the normal level. So normally the blood level should fluctuate between this 100 and 150, or when you're fasting it should fluctuate between 70 to 100. But when these fluctuations go roller coaster, the pink graph that I'm showing you. 1 minute, I'll just check if I can take a marker or something for better understanding. Just give me a minute. No. Not really. Okay, can you see my cursor if I'm moving it? Can you see that?
Yes, ma'am.
Huh.
We can
Yeah.
see the-
Yeah, that's all. So see this pink, and see the troughs and highs in the blood sugar. So this is bad. When there are, a lot of fluctuations, this is called as glycemic variability. And this glycemic variability increases in patients with diabetes and those who don't have glucose regulation or glucose tolerance. And this is the one of the main reasons which develops the diabetic complications. So it's not high sugar levels, it's the fluctuating sugar levels which is a problem. And of course, high sugar levels are, but fluctuating are worse than that. And pathophysiological mechanisms behind the complications are protein glycations, meaning all the proteins in the body, the glucose gets attached to that, and that leads to glycated proteins. And you must be knowing about glycated hemoglobin. Uh, HbA1c, that we normally term it as. So it's HbA1c, we see the hemoglobin because that is there in the blood and it's easy to estimate. But trust me, every protein in the body, it gets glycated, and that makes it a problem because this glycation leads to lot of reactive, oxidative, nitrosative radicals and pathways in the body. That's bad. So you must control that very well. Now, if I want to show you, I'm just trying to show you various diabetic complications like retinopathy, cardiomyopathy, nephropathy, peripheral vascular disorders, brain stroke, neuropathy, pain, super pain in the body, and diabetic food, that is gangrene. And I'm 100% sure, as physicians, you must have treated most of these conditions in your own capacity. But I'm just trying to tell you a different way to look at it from pathology point of view. So, how to go about it? Okay, now you have understood that these are the organs which are mostly affected by what you say, a high level of glucose. And if you see, all these organs are where glucose uptake is insulin independent. Meaning insulin independent, meaning whether insulin is there or not there, brain, heart, kidneys, heart, liver, brain, kidney, these organs will pick up glucose come what may, except liver, brain, heart, and kidney. All of them will pick up glucose come what may. And that's the reason these organs are the ones which suffer when diabetes affects a person. Now, this is a very simple experiment which is called as oral glucose tolerance test. So if you remember, I don't know how many of you have gone through that, but if you have to test diabetes in a person, normal person like us, so what they do is in the pathology lab, they either tell you to eat full and come after half an hour after the lunch, and they check your blood sugar level, and otherwise they give you sugar solution to drink, glucose solution to drink. So exactly the same way we do in animals, we give the animals a sugar solution to drink, and within 2 hours, 2 to 3 hours, we just try check their blood glucose levels, nothing else. Give them heavy dose of glucose and then check the blood glucose levels. So if it is diabetic, then you have this kind of a graph, and this is a normal graph. See, the green one is normal, the red one is diabetic. Now supposing the drug that you are screening, so all that you should do is you should make an animal diabetic, give him the drug first, then give him the glucose 75 milligrams in animals, you convert it to rat dose, and just check this graph for that particular drug treatment. If a particular drug is reducing the AUC meaning this level, if it is reducing the area under the curve of concentration versus time curve by 20% as compared to the diabetic control, then your drug may have anti-diabetic activity. You are not killing any animal, nothing. You are just giving a dose of, you are just making the animals diabetic and giving a dose of glucose to them. All those animals, you can use it further for your studies also. But this is the first step where you can understand whether your drug works or doesn't work. Okay. Now, these are two simple assays, alpha amylase and alpha glucosidase inhibition assays. Now, what happens, in the body when we eat starch, like starch in a sense, like when we eat chapatis or we eat rice, potatoes, or bread. So all this is starch. And this starch gets converted by amylase, a starch digesting enzyme, to glucose. And basically, before going to glucose, amylase converts it into maltose, lactose, disaccharides. And then the disaccharides are further converted into glucose by this enzyme, alpha glucosidase. Alpha glucosidase. So you can actually use these enzymes. These enzymes are available in the market, and you could just take a dietary polysaccharide, which gets converted into monosaccharides and disaccharides. And you could just take disaccharides and take alpha glucosidase and check the conversion into glucose. I'll just show you, meaning what I want to say is you could inhibit, if your drug, if your phytoconstituent inhibits alpha amylase or alpha glucosidase, then it could have an anti-diabetic potential. I'll give you a simple example. In modern medicine, there is one drug called as acarbose. This drug was invented in 1995, meaning very late. Insulin in 1920s, and then alpha amylase, alpha glucosidase inhibitor in 1995. So late, but then still this works. You don't need to have only insulin kind of an activity or metformin kind of an activity. These two activities are also easiest to study for a particular drug's evaluation of an activity. So simple experiment. All you do is you take substrate. Now substrate for amylase is starch. Just take any starch, corn starch, whatever. You take substrate solution. The procedures are available. And then you take sample solution. Sample solution meaning the drug which you want to test. It could be a suspension, because yours being a polyherbal formulations or Ayurvedic formulations, it could be a turbid or a suspension also. So mix it with that And for a positive control, you can take acarbose because acarbose inhibits. And then you pre-warm it, meaning make the conditions conducive to the body conditions. Add the enzyme solution. So you already have a substrate there and the drug, and you have pre-warmed it at 37, and now you are adding the enzyme solution. Mix it and incubate it for 30 minutes. So 30 minutes you give the enzyme to work on the substrate. If your drug is going to inhibit the substrate, you won't get the final product, meaning the final product will be less. So that's what you can understand by doing this assay, amylase assay. One sec. Yeah, sorry for that. Okay. So what I was trying to tell you is that if there is a less production of enzyme, which you can detect by using a indicator, this is a very good technique for understanding whether it inhibits amylase or alpha glucosidase. And you could definitely understand the product conversion. If the product is less, your drug is active. So you've done two assays. One is glucose tolerance test in whole animals, and this is in completely in vitro buying enzymes. And in fact, sometimes when if the enzymes are expensive, there are many times animals to be sacrificed. So we just take intestinal pieces of the animals and we homogenate those pieces, which has got enzyme in that. And then it becomes cheaper if you have animals with you. If not, then buying enzyme is the best way, and you can do many samples at a time. So this was about the amylase inhibition. Now, something very interesting, like if you have hyperglycemia, if you have high sugar level, you know that the cellular glucose levels go up, particularly in those areas where the sugar gets absorbed without insulin, meaning heart, liver, heart, brain, kidneys, and it gets into the mitochondria, the cellular suborganel organelle. And then in the mitochondria, the glucose is utilized to make ATPs. But when you have lot of glucose, mitochondria produces whole lot of reactive radicals. And these reactive radicals they result into many pathways. What are these pathways? Polyol pathways, advanced glycation end product pathway, protein kinase C, and hexosamine pathway. And all these pathway activation leads to all the problems which we saw: retinopathy, cataractogenesis, cardiomyopathy, etc. So do you think by stopping glucose, meaning हम लोग बोलते हैं ना insulin दे दो, metformin, you give metformin and reduce the amount of sugar, will you be able to control all these problems? Is it possible to stop all these? Answer is no. Answer is no. That is because the mitochondria, they have metabolic memory. They know, they get into that mode of producing reactive radicals. So that is the reason antioxidant drugs, they work very well as far as all these diabetic complications are concerned. And you will also see that antioxidant drugs are really useful in treating many of the inflammatory conditions also. So anti-diabetics, meaning reducing the sugar, yes, you can. But don't worry if it doesn't reduce the sugar, but it has got antioxidant or anti-inflammatory activity, it could work in treating the diabetic complications, is what I want to say. Okay, so I'm not getting into the details of this simply because you are doctors and you are not chemists, but I'm just trying to explain you the chemistry that when the glucose combines with protein at every different stage, it produces reactive oxygen radicals. While it produces advanced glycation end products, it produces reactive radicals at every stage. An example of advanced glycation end product I've given you is the HbA1c, which is there in the blood. That's an age product which we are able to detect in the body. So this is a reactive oxygen stress which leads to diabetic complication. So a very simple experiment you can do in your lab if you have a spectrophotometer in your lab. So I'm just explaining the whole slide first. Okay. So what we generally do now, we take albumin, simple albumin. You can buy albumin, mix it with glucose solution. The protocols are there, and incubate it in the in incubator at 37 degrees. And slowly, you'll have all those 4 stages which we saw. There is a Schiff's base here. See, there is a Schiff's base, amidori product, dicarbonyl products, and ages. So at the end of 4th week, you'll see age. It's like 1 week, 2 week, 3 week, and 4 week. So every week you can see that. And you can add your drug to be evaluated in the same medium, albumin plus glucose plus drug. And you could see whether the end product age is reduced or not really reduced. It's a simple spectrophotometric assay. So this is a compound in methi seeds. Naringenin is a compound in methi seeds, which we tested it against age formation. Simple experiment, as I told you. And this naringenin decreased age formation. The final one is decreased the amidori products, meaning a stage which leads to dicarbonyl compounds also. It increased the thiol content. So if you are aware that in the body there is something like glutathione. So glutathione is responsible for scavenging reactive radicals. So naringenin could even increase the thiol content of the meaning in the experiment it could increase the thiol content. That's what we saw. It is measurable, and it also decreased protein carbonyl content. So this is such an interesting story. We all know that methi seeds are good for treatment of diabetic complications, but then why is it so? That it is because of the naringenin. So this is one aspect of it. But supposing if you have any other drug to be tested, you can test it in the similar manner is what I want to tell you. Very simple experiment. No, not much money involved in that. So this is a third experiment which I showed you, which you can do for evaluating anti-diabetic potential. Now, the next is age and rage interaction. Why do you say that age is a problem? Meaning age, meaning advanced glycation. Why do you say so? Because whenever the body has ages, whenever the proteins are glycated, the macrophages, meaning the blood cells which are responsible for providing us with innate immunity, they express an receptor called as RAGE, R-A-G-E. I'm sorry. Yeah. So this, whenever there is age, the macrophages as well as the endothelial cells meaning the cells lining the blood vessels, both of these cells, they are capable of expressing a receptor for age. And when they express this receptor for age, that's a bad event that happens. So when they express this age can come and immediately bind with the rage. And this combination is bad. Age-rage combination, it activates the inflammatory pathway, MAP kinase and P21RAS pathway. And this leads to inflammatory gene activation, which is NF-kappa B. This is in the cell gene, cell nucleus. And when NF-kappa B is activated, TNF-alpha, which is a inflammatory marker, that gets activated and some interleukins. And while doing that, it also generates a lot of reactive radicals, meaning age-rage combination has done this deleterious effect here. And then this TNF-alpha further produces more oxidative stress, of course, inflammation, and that leads to diabetic complication. So another aspect of looking at it is, can your drug stop this age and rage binding? Can your drug not allow the age to bind with the rage? Just let us try and see this, whether we can do it. So we can do it. And this is little expensive because you need that setup for doing cellular assays. So these are the macrophage cell lines. You get that. And these cells are grown in 96 well plate. And you grow them. And once you grow them, you add age to them, meaning try and settle them, and you add age to them. Age meaning the one albumin glucose combination that we have made. The same thing. You can add age to that and keep it for 1 hour. So when you keep it for 1 hour, the cells will express rage receptor for age. And now you have to identify how much of the you have to identify the interaction between age and rage. So again, you add an extra age to that. And but this time the age is combined with a dye, fluorescent dye, FITC dye. So you add this alone or in presence of your drug to be tested. So if your drug does not allow the age-rage complex, the age will not attach to the rage now because you're adding extra age. First you have added age and then the rages are expressed. Once the rages are expressed, you are adding labeled age to that. So if your drug doesn't allow that to bind, you won't have fluorescence. If it binds, you will have fluorescence. So you incubate it along with the drug for another 1 hour. The tagged age with the drug, you incubate it for 1 hour. Keep it for 1 hour and then wash, meaning the cells get attached. So the experiment is over. If there was a binding to rage, the experiment is over. All the extra labeled age, extra drug, everything you'll wash it out. And after you wash it out, to the cells which are attached here in this plate, you add triton. Triton is a surfactant. So this would break the cells and this would release the FITC dye, meaning release meaning age, rage, whatever combination is there, that dye will be given out from the cells and you can measure it at 495 to 520. And it's such an easy. Hello. I can't hear anything. Do you want to say anything? Okay, I think I'll go ahead. There is some disturbance. Please mute. So what I want to tell you is that thymoquinone at 100 micromolar concentration inhibited this age-rage interaction. So this thymoquinone is from ajwain, ova. So I'm just trying to tell you that we are just trying to explain why ova could have an anti-diabetic potential, why methi could have anti-diabetic.
Hello,
Yes.
I'm sorry to interrupt you.
Yeah.
Dr. Ajay Ratna, please mute yourself. Now you continue, ma'am.
Yeah. I think I was good enough. Meaning, have you understood this particular thing? Then I'll go ahead. Otherwise, yeah. I think we can go ahead. We can take questions at the end.
Yes, ma'am. Okay, ma'am.
I don't know why it is not going ahead. I'll just stop share and share it again. 1 minute. It's got caught.
Okay.
Yeah, I'm sorry for that. Yeah. So we are here. Now so that's what we did. Like we selected, I think this is the end of the presentation probably. We selected the phytoconstituents, we did the study. Oh, one more thing remains is animal study. I'll just go ahead with that. So now I just want to share one of the experiments which we did it on this particular flowers, and these are called as Sita Ashoka flowers. They look like Exora, but they are not Exora, and literature reported that it is useful in diabetes, cancer, uterine disorders, etc. And we now, I'll explain you how we did the polyol pathway. So there is one more simple pathway which is called as aldose reductase pathway. And generally when you have normal sugar, there is only glucose gets converted to glucose 6 phosphate and then to ATP. But when there is hyperglycemia, when there is more of glucose, then the aldose reductase converts it into sorbitol and fructose. Now this sorbitol is very very osmotic, it can pull water towards it, and that's what happens in cataractogenesis. This aldose reductase pulls a lot of water and changes the orbital pathophysiology, and that leads to cataractogenesis and stress in the eyes. So normally aldose reductase is only 3% active, and when there is high sugar it becomes 10 more times active. So aldose reductase could be one of your targets. That's what I'm trying to say. Along with amylase, alpha glucosidase, anti-age, aldose reductase could be one of your targets. So what, it's a very simple experiment. You take rat eye lens, and these days I even collected from Devnar abattoir. So Devnar abattoir, every day daily they have goats being sacrificed, cattle being sacrificed, so you'll get lot of eyes, and you can get aldose reductase enzyme from those lenses of those eyeballs, and you just homogenize that, and so you have your aldose reductase enzyme. This is a very simple procedure where you take the fluid from that eye homogenate, which contains aldose reductase, and you work it out. You work it out using a, so your enzyme is aldose reductase, the substrate is xylose, because you need to have some substrate, and aldose reductase works on xylose, and then that converts it into glucose, and that's what we will be studying in this particular experiment. Very simple experiment. So the way I showed you about the amylase and alpha glucosidase, similar example, but where is what enzyme you will take? Either you will buy aldose reductase from the market, or you will make your aldose reductase, and you will take xylose and work it out in a small Eppendorf tube. So very less amount is needed, and you can do as many phytoconstituents as you wish to do, and spectrophotometrically you can measure. So we actually worked on, 1 minute, yeah, on different extracts of that Saraca indica. SI meaning Saraca indica. So methanolic extract, ethanolic extract, butanolic extract, pet ether extract, water extract, and standard we took quercetin as an antioxidant. And we did the same assay like alpha amylase assay, but with aldose reductase. And we saw that quercetin is equally, like ethyl acetate extract showed equal activity like that of quercetin, and meaning almost similar inhibition of aldose reductase. That means similar activity on diabetic complication of the eye, that is cataractogenesis. So let us study this, whether we can do this in animals for that matter. Now we know that ethyl acetate extract works in vitro. So let's see if it works in the animals also. So the first what we did, first we got goat lenses from the Devnar abattoir, and we exposed them to high glucose levels, and literally cataractogenesis is formed in the Petri plate. Don't even have to use animals, and you don't need any ethics permission for this. It's a very simple experiment. Only thing that you have to remember that every day you have to change the glucose solution. Otherwise, meaning keep the same 55 millimolar glucose. But what happens, with the glucose, there are chances of having fungal infection. So this is a challenge in optimizing this assay, but it is easy. That's not a meaning difficult thing. So I'm just showing you the representation that this is the normal lens where you can, count the number of squares. This is a cataract lens where it was only high glucose solution. In this, it was quercetin plus high glucose solution, quercetin 1 milligram per ml. Here it was ethyl acetate extract 0.5 and ethyl acetate extract 1 milligram. And you can see that 1 milligram is similar to quercetin. So similar to quercetin, not exactly same. So this is what a simple experiment you can do. And more than that, you can also evaluate the antioxidant enzymes and proteins in the lens protein. These are also simple spectrophotometric evaluations: superoxide dismutase, catalase, and reduced glutathione. So reduced glutathione is the thiol. So more is the scavenging, and superoxide dismutase increase it is better is the activity. And so you see all these enzyme levels increase with ethyl acetate extract of Saraca indica. So that means what? It is an antioxidant and it is an aldose reductase inhibitor. And you have also seen the anti-cataract activity of the same. So you have proven that ethyl acetate fraction has the capacity. Now we went on to animal study, main study. So before you're sure that your drug works, you shouldn't go in for animal study because that's unnecessarily expensive. So every week, so this was the study. First of all, diabetes was induced in a male Wistar rat, and ethyl acetate extract 200 to 400 milligrams per kg orally was given to rats, and blood glucose was measured on 0 and 40th day. Not only on that, we also did it in between every week because it's simple. We have a like if you have a glucometer, and we had eye scoring observation every week. So we used to call a veterinary ophthalmologist to check that. And we did at the end of the study, that is at the 48th day, meaning 7th week, we did the aldose reductase estimation of the eyes of the treated versus non-treated versus different doses treated animals. And this is I'm just showing you the final slide. A lot of it goes into it, but this is just for the showing you that this is the normal eye. And see the ESA treated eye, 400 milligrams per kg treated eye. 200 milligrams per kg treated eye is opaque, like that of cataract. But 400 milligrams is almost similar to the normal. And these are the duration and these are the stages of cataract. So you can see that 400 milligrams treated, it really comes down very low. I mean, you really don't see only any cataract stage for that matter. Okay. So we did glucose levels in those animals. So if you can see, glucose levels were not reduced by ESA, meaning by the ethyl acetate Saraca indica extract. So what? It doesn't, but it reduces aldose reductase considerably. So, and it reduces cataractogenesis considerably. And so we proved that this bioactive fraction of Saraca indica, it prevents cataractogenesis, which is mediated through aldose reductase inhibition. So the thymoquinone paper that I showed you is here. Divya Kanchan did that. And Gaurish Somani did this aldose reductase inhibitory activity and animal study. But let me also share it with you. Recently, we filed a patent and we are almost through with the patent, meaning the final hearing is also done. We are just waiting for the National Biodiversity Authority. So now what we did, see, the dose of ethyl acetate extract was about 200 and 400 milligrams per kg orally. So we don't want to waste that. We made eye drops of that ethyl acetate extract, and that we patented. And it's working wonderfully. Like further studies are going on. I'm also looking out for partners where we can, uh, take this drug to public, and that I have not seen any drug which is used for cataractogenesis as of now. And we have done a full-fledged animal studies with eye drops also. So it's easy for the patients to take it over a long period of time. So this was about my research. This was about explaining you. So Akruti worked on diabetic nephropathy. So we have worked almost on every aspect of diabetes mellitus, and this is the current team who are working with me. The top line is all PhD students, and the bottom line is all master students. So now, if you have any questions, I'm happy to take them on.
Okay, Ashish sir.
I'll just check out in my. Chat, I don't see any questions out here.
Hello.
Yes, ma'am.
Thank you very much, ma'am, for throwing light and sharing your knowledge. Now I invite Dr. Ashish Kunde for question and answer session.
Dr. Ashish.
Thank you, ma'am.
Dr. Ashish?
Participant, if any queries regarding today's lecture, please put it in the chat box.
I can take it late. I mean, are you okay with the time? Because yeah, I think I'm okay.
Yes,
Yes,
I have
ma'am.
always have a habit of going beyond time.
No, it's okay.
No, I don't see any questions in the chat box as of now. I can take it later also. I mean, you
Yes.
could send it through Madam Sheila, and I will
Yes.
answer
yes,
them.
ma'am.
If you would like to, you can also extend your lecture, ma'am, if you would like.
Extend.
Yes, if you would
No,
like to.
if anybody wants to, and currently I'll just tell you, see, Saraca indica eye drops, so we patented it, but right now what we are doing is we are even trying to see whether the actives in Saraca indica we can make eye drops of that.
Yes.
So we have quercetin and
Very wonderful and very novel work, ma'am.
even that's what I feel. Ayurveda has got so much to offer, and you people can really work. If any of your students wish to do any research, it's a lot of
Okay, in
things.
collaboration.
Yes, we can do it in collab. Ideas will be yours because
Yes.
we don't know much about Ayurveda, but I know Ayurveda is a really great science.
Okay, thank you.
Okay,
Okay, ma'am.
thank you. And I can take questions later on. So right now I'm just leaving the room for that matter.
Ashwini, ma'am.
Yes, ma'am.
Okay. Thank you, madam. Thank you.
Yeah. Thank you so much.
Such a nice lecture, extensive lecture. Okay, thank you.
Thank you.
Ma'am,
Okay, now
can
I
I
will
ask a question?
Yeah.
Now they are asking queries.
No, it's okay now. I have kept time for them.
Yes. Yes, parents
Ma'am,
can be asked.
ma'am, you have already explained the toxicological assay, but can I know the details of it, like why it is needed for the Ayurvedic drugs, where already the dosage and the duration for the duration is prescribed for the specific drug?
Yes. See, I'll tell you why. That particular drug that I got now, it was for increasing the aphrodisiac activity. So what they said that you give it in male rats over a period of time. Of course, male and female both they had taken. So they felt that if you give it for a long time, whether it has got any effect, like if they prescribe for a human being to take it over a long period of time, is there by any chance any toxicity? Because see, when you want to export such drugs, and when you say that there is arsenic in this amount and mercury in this amount, which is there, but I'm 100% sure that Ayurvedic drugs they also have the processes done very well. But sometimes some companies, I don't know, maybe they don't make it well, they don't formulate it well, they don't follow the Ayurvedic text well, and then the side effects are seen for Ayurvedic drugs. And so there is a flack, there is a, what you say, globally people feel Ayurveda, oh my God, kidney toxicity. That's what they feel. It's not that. So they wanted to prove, madam, do it and show it that it is not there. So a complete protocol was followed according to Schedule Y guidelines, Indian guidelines, and the study was done.
Thank you. Thank you. Next, any query? Queries are welcome. Students, PG students, our
So generally, what do the students study, like do research on?
there are different area. Our clinical students work on these aspects, diabetes mellitus and all, and our students also. But drugs are formulations, Ayurvedic formulations, which are mentioned in our classical texts.
Correct.
They also do animal study, madam. In future,
Okay.
if we wanted to, we wanted to seek your guidance,
Sure,
we'll
sure. No
certainly
problem.
contact you, ma'am.
No problem. No problem.
Next, anybody query?
Ma'am, last query, please. Can I ask?
Yeah,
Yes.
please.
Ma'am, before going to the in vivo toxicity study, is it necessary to conduct the in vitro toxicity assay, cytotoxicity assay?
In fact, it's easier to conduct that. And nowadays, even zebra fishes are being used. That's also quite cheap. And animal toxicity studies would require ethics permission also. So, cellular and zebra fish is better than animal toxicity. That's what I feel. Basic idea you get. But that is for medications which are not already proven. Meaning, how I told you, this particular aphrodisiac drug, they wanted to give it in patients. So they just wanted to show that animal safety data is very good. There's no problem. So even if you show the cellular data and they say then show it in animals. It generally happens. I'm talking of exporting a particular formulation. Marketing license to everybody gets, meaning if you show whatever data that the regulatory agencies expect. Apart from that,
Further, anybody from students, postgraduate students? Okay. Ashish sir, shall we stop? Yes.
Yes, ma'am.
Okay, ma'am. Thank you very much, ma'am.
okay, thank you.
Hello.
Madam has left, I think.
Hello. Now
Hello.
I declare
You
first
announced
session
that.
is over.
Hello.
And I request all of you to please rejoin by sharp 2 p.m. again. Thank you once again.
Hello everybody, this lecture will be available on YouTube lifetime. So please share, subscribe and like our YouTube channel, YMT Ayurvedic Medical College and Hospital. Thank you very much.
Ask them to rejoin also at 2 p.m.
And please rejoin in next session on 2 p.m. Thank you. Hello, Pramod sir.
आज